Herman Merkel pressed his palm to the bark of an American chestnut at the Bronx Zoo and felt something wrong. The tree was oozing, its bark split in jagged seams, the wood beneath turning soft and dark. And this was not how a giant died, not the tree that built barns, fed bears, darkened autumn hills with spiny green burrs. Merkel was the zoo's chief forester. He knew sick trees. He told himself it was the heat, a few bad specimens near the new ornamental plantings from Japan. Nothing that could touch the millions of chestnuts standing across the Appalachians, each one 100 feet tall, each one the backbone of a forest from Maine to Georgia. But the fungus had already arrived. It had ridden silently in the bark of those Japanese chestnuts, planted years earlier as decoration. And from this single garden, it would tear outward at 20 to 50 miles a year. 4 billion trees would fall. The greatest forest collapse in modern North American history, set loose by a gardener's choice of pretty greenery. And when the cornerstone vanished, what would hold the forest together? The first wound was already weeping. Merkel cut samples from the cankered trunk and sent them to William Murrell at the New York Botanical Garden. Murrell was a mycologist, a fungus hunter, and what he saw made him publish fast. The invader was Cryphonectria parasitica. A spore landed in bark, threaded through the cambium, the tree's living pipeline, secreted oxalic acid. Tissue died, swelled into cankers, girdled the trunk, choked water and nutrients until the crown starved while still standing. No cure, no pruning deep enough, no chemical that reached. Wind carried it, rain splashed it, birds brushed weeping wounds and flew on. From that single garden it rolled south, through Pennsylvania, through Virginia, down the Appalachians. By the 1920s, ridgelines showed the wave, whole mountains grey with standing dead. And beneath those skeletons, hunger was waking. The nut rain had never failed. What would the forest do when it stopped? Across 200 million acres, chestnut branches bent under a crop so reliable that black bears timed their litters to it. Passenger pigeons darkened the sky in millions. Deer fattened, turkeys scratched. 1 in every 4 hardwoods was a chestnut, and settlers built barns from wood that outlasted generations. Then the rain stopped. Bears emerged lean from their dens. The great pigeon flocks found hollow branches where abundance had been. Insects that lived only on chestnut pollen simply vanished. What Murrell had named was not a tree disease. It was the deletion of a pillar from a structure with no redundancy. 4 billion pillars, oaks and hickories, rushed upward into the sudden light, but oaks masked in boom and bust cycles. The forest looked green again. It was not the same forest, and something stranger was waiting underground. Decades passed. Oaks filled the canopy gaps. The forest looked healed. But plant pathologists who dug into the dirt found something wrong. The fungal networks that had partnered with chestnut roots for millennia, trading minerals for sugar, weaving threads through the soil, were diminished or gone. Oaks and hickories had brought their own fungal associates. They were not the same. The soil chemistry had shifted, nutrient cycles had changed. Then researchers began pulling up buried logs, fallen chestnuts pressed into earth for nearly a century. Still carried fungi from the pre-blight forest microbes that had colonized the living wood, preserved like messages in a bottle. The forest floor was haunted, not by ghosts in any storybook sense, but by the absence of a species that had engineered the very dirt beneath the new canopy. The wound ran deeper than the eye could follow. And if the ghost of the chestnut still lives in the dirt, the question is no longer whether it survived, but what it's been doing down there all this time. Above ground, the chestnut kept trying. In clear cuts, along old fence rows, on steep Appalachian slopes, green shoots pushed through leaf litter. These weren't seedlings. They were children of ancient root systems, some older than the blight itself, still alive underground where the fungus couldn't reach. Every spring, billions sent up slender stems, grasping for light. Within a few years, sometimes sooner, the blight found them. Cankers formed, bark split. The shoot died back to the collar, and the root retreated, gathering strength for another try. The American chestnut wasn't extinct, it was trapped. A species caught in eternal adolescence, growing up only to be cut down, again and again. The genetic memory of 4 billion trees still pulsed in those roots. But it couldn't cross into the canopy where a forest tree belongs. The species was caught in a loop, reaching for the canopy, cut back, reaching again, and no one knew whether the loop could ever be broken. Plant geneticists looked at those struggling sprouts and saw what no one else had. Not ghosts, a living library of wild American chestnut genes, and the raw material for a comeback. The roots had survived because they sat below the fungus's reach. Every sprout carried the full blueprint of a tree that had once ruled 200 million acres. But a blueprint is not a shield. The question was how to arm it against an enemy it had never met. In 1983, corn geneticist Charles Burnham proposed an answer. He founded the American Chestnut Foundation and launched a back-cross breeding program, crossing surviving American chestnuts with blight-resistant Chinese chestnuts, then breeding the offspring back toward the American type again and again. The goal was a tree 15/16 American, with just enough foreign DNA to survive. Meanwhile, at the State University of New York, researchers William Powell and Charles Maynard took a bolder route. They engineered a line called Darling 58, inserting a single wheat gene that makes an enzyme oxalate oxidase, which breaks down the oxalic acid the fungus uses to kill. The tree neutralizes the invader's main weapon. For the first time, a blight-resistant American chestnut is within reach. The species stands on the edge of resurrection. But a lab-tested sapling is not the same as a tree standing in the real forest, facing wind, drought, and a century of altered soil. When researchers split open a chestnut log buried since the 1920s, they found fungi inside that had vanished from the surface world generations ago. The log had become a time capsule. But the deeper surprise came next. Ancient chestnut stumps still push up sprouts today, and those dying shoots still host ectomycorrhizal fungi in the surrounding soil. The fungal web had not forgotten its partner. It had simply been waiting. This changes what restoration means. Planting a blight-resistant chestnut is not merely adding a tree, it is dropping a key into a lock that has sat rusting for a century. The soil remembers. The fungal partners remain. The frame of the old forest still hums beneath the oak-hickory canopy that replaced it. A genetically modified sapling, planted where its ancestors fell, could reawaken networks that predate the blight itself. One engineered tree dropped into soil that remembers may be enough to start. But stitching back a world that took millennia to weave is a longer story than any single planting. One sapling in Virginia holds part of the answer. Its leaves carry a wheat gene that disarms the blight's acid weapon, and its first root hairs push into soil that cradled fallen ancestors a century ago. Somewhere below, dormant fungal spores stir. The tree that died standing is finally getting its chance at the canopy. But the forest didn't merely lose a tree. It lost an intricate world that reached from canopy to soil, a web the chestnut had stitched together over millennia. That web never fully disappeared. It went dormant, preserved in buried logs, in stubborn root sprouts in the soil's own memory. The century of silence wasn't an ending, it was waiting. Restoration isn't about bringing back one species. It's about giving an entire ecosystem permission to remember what it once was and to become whole again. When this sapling's roots meet the waiting fungi, when its canopy finally opens to the sky, the oak-hickory forest that grew up in the chestnut's absence may finally stand taller than it has in generations. The key's been found, the lock is turning. If stories like this, where hidden connections beneath our feet prove more resilient than we imagined, make you want to keep looking closer at the living world, subscribe, and we'll keep walking these trails together.